Hello, we're live. We did it.
Finally.
Yeah.
Yeah, long time coming.
If you could see all of the equipment that's on this table before us, you would be shocked, I believe. But this is the Science Club show. I'm Ross Balch, and I'm being joined this week by Tina.
Hi, everyone.
And Ben Kiernan.
Hello.
It's an absolute pleasure to be live doing this. I've had this idea in my head for a long time to do this science show that's live with a lot of audience interaction. You'll notice we're on Twitch today, which is an excellent platform for interacting. You have a little chat to the right of the screen, and we'd love to get your comments and questions during the show. We are here basically to interface with the public, essentially, and make science communication more of a dialogue than... I think a lot of science communication is a lot of being talked at, but we want to talk with you, essentially.
Yeah, absolutely. The whole purpose, I believe, of the Science Club show is to, well, it's two things, I guess. We want to sort of make science a lot more digestible and more accessible to the public, because a lot of people sort of have the perception that science is a little dry, it's not so engaging, and it's a little bit opaque, I guess. It's hard to get into. But we want to sort of alleviate that by initiating the discussion with you. And yes, so please come over to our Twitter, come over to our Twitch, have a chat, have a conversation. And we're keen to hear what you think.
Yeah, so I've just noticed that Ben has sort of disappeared, so I'm going to keep talking. And while I get you on screen, Ben, why don't you tell our audience about yourself, and I will make sure that you are not a little blob, essentially.
Oh, okay. Well, hopefully I will appear shortly. I am Ben Kiernan. I am a science communicator working currently in a PR firm that focuses on science. I have a background in physics, but recently I've been hosting the non-peer-reviewed podcast, which is a mostly weekly SciComm podcast. It's a light-hearted look at some of the most recent science news, and it's myself and two guests. Ross is one of the guests on the show that appears very often on the show, and I'm very grateful for. And we look at some of the most recent science news, we answer some questions, and we have some fun.
Yeah, we do. I think that's one of the nice things about the peer review podcast. It's a very much a light-hearted look at science. And one of my favorite sections is where Ben, he will troll the internet for some of the... Initially it started as like dumb questions about science, but actually it's kind of turned into just sort of different and interesting questions about science that we can somewhat speculate about what the person was thinking about when they asked the question, and also just a little bit talk about some science. And yeah, we've come up with some surprising topics in that regard as well.
Yeah, I still do find some dumb questions on there, but they have some interesting science behind them we can talk about. So the non-peer-reviewed podcast is our name and our disclaimer.
Yeah.
So we have fun, and everything we say on the show should not be quoted professionally.
Yes, absolutely. So, Tina, why don't you tell people about why you're here on the show?
Well, basically, I've been dragged into this by Ross Balch, mainly because we're actually housemates, but we've also been working as scientists in the same building. I'm doing my PhD, and I believe Ross is working for a infectious disease group.
Yeah.
We mostly became friends mainly because of football, and then we discovered a love for Magic the Gathering.
Yeah.
But first and foremost, we are both always scientists, and not just about the lab side of things. We are more interested in getting science out to the public.
Yeah.
And we both always had a passion for that. And I guess, this is perhaps the best way that we can to do it with the instruments that we have. So let's hope for the best, I guess.
Yeah, I think we all just here, like, obviously we have done science. Like Ben, of course, you're working in communications now professionally, but you obviously have undergraduate degrees in science, and you've done a research project as well, so you know what research is like. And I think that's a nice thing for communicators to be able to bring to the table, people who've done research and sort of know about what science is really like. I think in the media sometimes there's this idea that every time some new discovery happens, it's the new definitive piece of knowledge that we have, when I think we would know that oftentimes it's a little tiny step and it's a little tiny piece of evidence that's really interesting, but sometimes you have to be a little more discerning, I think.
Absolutely. In fact, like you mentioned, everybody sees the big discoveries that happened as advertised, people winning Nobel Prizes and things like that. But if you actually do listen to the speeches that they give, everybody is aware that science is built on the shoulders of a lot of people. In fact, it's like you said, it's an incremental process. There's a lot of people contribute to every big discovery, right? It's just that one person happens to make the breakthrough or a novel idea or just a unique way of looking at the data or what you're working on, and therefore they make the leap. But without the contribution of all the people, the hundreds of people that come before them, that may not actually be possible.
Yeah, exactly.
Yeah.
But also, science is just a lot of fun, isn't it? I think when we did this like little pre-show, I was telling about a story where, when science really first started existing, which, to be fair, the idea of science as it really has solidified as a method has only really existed now for 400-500 years. Like really the time of Newton and those people, they were the ones who were really first starting to do the scientific method in terms of writing observations, coming up with hypotheses, trying to falsify those hypotheses. And there were a lot of philosophers and poets at the time. They were a bit unhappy with science. They didn't like the idea that you could look at a rainbow and rather than just appreciate its beauty, you could explain it. The idea of refraction of light through water in the air, the fact that you could explain how this happened, for them seemed to trivialize or take away the beauty of something, whereas for me it's the exact opposite. I think the great thing about science is that when you understand things, you can truly appreciate what they are. I mean, imagine the rainbow example. It takes so long. Like, imagine how much had to go right for a rainbow to appear. The sun being in a specific version of the sky, light rays from the sun traveled 8 minutes to our planet. They went through raindrops that had to be oriented in just the right direction for the refraction to be perfect. Like, it's crazy to me when you think about the complexity of life in the universe, how anyone could know about that and somehow see that life is diminished because you took the mystery away from it. I mean, science doesn't really take the mystery away from anything. It just creates several new ones.
Absolutely. We were just talking about this the other day, and I used the analogy of a magic trick, for example, because a magic trick is interesting as long as the mystery remains. And then, so some people, I guess, perceive that when you take the mystery away from the magic trick, it becomes something inane. But at the same time, I guess, if you were a magician yourself and you saw somebody else do a unique magic trick, like you would appreciate not the trick as much, but the manner in which it was done. And hence, I guess, currently, as it is, scientists probably would have a better appreciation of science. But, that's the reason why we have science communicators, really, because we are trying to get people to understand the fundamentals of science much better than they already do. So at least, in that sense, they would be able to see things, however elementary, from the perspective of a scientist, and therefore appreciate the beauty of science more, perhaps. What do you think, Ben? I guess since you work in the space, is that what science communicators do in general? Or what are you trying to achieve? Yeah.
I would say in general, science communicators spend a lot of time reading papers that are very hard to digest and then trying to make those digestible to the average population. And like there is, as you said, a lot of great science being done out there. But for the most part, people aren't hearing about it. And so like that's kind of what science communication is doing. It's sharing the discoveries that have been made with everybody else. Everybody outside of that science bubble needs to know what cool things are being done inside that science bubble. But often scientists are so wrapped up in the process of science and the academic publishing cycle that needs to keep happening to make sure you keep being employed by universities and research organizations, that they do the research, get it published, go, great, have a glass of champagne, back to the research, keep doing stuff. And then there's an extra step that doesn't get done as much as it should, which is talk to a media comms team and then tell other people that science. So a good example was last year, a paper came out by Macquarie University, led by Richard de Gregis, a physicist, proving that the Milky Way is warped and twisted. So rather than the flat disk that we all thought it was, it's actually more Pringle shaped.
Okay.
And this is something that astronomers sort of knew, but that hadn't been published and it hadn't been spread out through to the rest of the community, to the wider populace. And that story did fantastically because, Milky Way is warped and twisted is a cool story.
Yeah.
But no one had told, everybody else.
Yeah, exactly. So yeah, that's why we're here. We just want to talk about science, and hopefully one of the nice things is that sometimes when you are into a subject, you don't always know what your audience doesn't know. And as a communicator, you're trying to guess the kind of things that you want to explain without trying to over-explain. Because, you don't want to treat your audience like idiots. People are smart, I think, for the most part, and can work a lot of things out for themselves. But of course, you don't know until you talk to people. And that's what we're hoping we can achieve with this show. That, if we're explaining something and if we're not doing a good job and you don't get it, then that's great. We can get this instant feedback because of Twitch, and we can explain things hopefully in a really in a better way the next time, which I think is really important. So I guess we should start talking about some science then. We've talked about ourselves and our passion a little bit long enough, I think. So obviously...
Wait, hang on a minute, Ross. Have you talked about yourself?
Oh, good question, Ben.
So I guess I did talk a little bit about myself at the start, but then handed it off. But yeah, so I'm an infectious disease researcher. That's kind of my bread and butter. I've really, ever since undergrad, doing the sort of molecular, like microbiology courses, infectious disease is what really took my interest. At the time, it was antibiotic resistance, and then I moved on to doing more things about viruses, because viruses are kind of unique in the world of life in that it could be argued that they're not even living organisms. They're actually just replicating biological machines, and they have the smallest amount of DNA out of any creature on the planet. And again, creature is a debatable word, but they do so much, and they can achieve so much during infections. And we're really going to be talking at length about that in just a few minutes. So that's me. And I've just had a passion for science communication for a long time, and this idea has been in my head for a while, and now I have the resources to do it, essentially. You don't, as the audience, you can't see all the crazy behind the scenes gear that is making this happen. So it is going to be...
Hopefully soon you'll post a picture of your light boxes, because those light boxes they're MacGyvered. They're great.
Absolutely.
So basically, to get this to happen, we crafted things that would normally cost thousands of dollars out of things you can buy at Kmart.
Pretty much, yeah. And Bunnings, don't forget Bunnings.
Yeah. So I have a comment here from Madam Zelda who says, one problem, in my opinion, is that schools are still teaching so many things that are out of date. So lay people argue thinking they have knowledge on their side, but it is out of date. And this is actually a really good point, because even as an undergrad, this is still a problem. I learned things as an undergrad that when I became a researcher, I realized people had stopped using that technique 10 years ago. And also, I think, when it comes to schools, another interesting thing is with the way science is taught is that science is taught as this nebulous object of things where it's like, this is what we know. This is a fact. But science doesn't deal with facts really at all. Science doesn't even really deal with what we know. Science deals mostly with what we can rule out.
Yeah,
which
absolutely.
sounds weird, but that's what science is about. It's essentially about ruling out every possibility until you have the one thing that is the most likely to be true.
Yep.
But we see this all the time, that things can often turn out not to be true, even after a long time.
Yep.
Something that's been happening recently is we're all kind of aware, as Australians especially, because we have a Nobel Prize winner in this field, is the idea that the universe is expanding, and that the rate that the universe is expanding itself is increasing all the time. And that's based on observations of these supernovae that have a very particular brightness when they go off. So obviously we can tell how far away these happened because of how bright they look to us. And there's some evidence now that maybe as we trickle in, some people are looking at evidence that maybe disproves that even though that evidence was really strong, and we'll see how that goes in the future as well. So I think
so one of the examples I come up with is I have a physics background. So you go through your entire high school and then the first year of undergraduate, and you're getting taught, like, here are the equations, here are the next step, here's the next step. And then you get into second year physics and they say, okay, all these things we've learned, they've just been stopgaps. They've been like simplified versions of everything. Now we're going to teach you what's actually going on, because we weren't taking into account relativity or quantum mechanics or anything that's actually going on. When you get down to it, physicists barely understand how gravity works. But it's so fundamental to so many equations that we figured out the closest thing we can, and we just use that because it works for most cases. And so as you go along, the best example is if I'm traveling on a train and I throw a ball, the simplest solution is, well, you add the speed of the train and the speed of the ball. The ball is now traveling 60 kilometers plus 10 kilometers, it's now 70 kilometers.
Sure.
And that's why people think about speed of light. If I'm traveling close to the speed of light and then I shine something at the speed of light ahead of me, do they add together? Is it now 1 point something the speed of light? Is it 2 times the speed of light? Well, that's because they're using, school level physics and not taking into account the higher level, because there's a whole extra level to that equation that people learn in school that just you don't learn unless you do it in university.
Yeah.
And I'm glad you brought out physics as well, Ben, because I just wanted to mention about the discovery of the Higgs boson. But I guess the point that I was trying to make is that we are sort of limited by the technology that we have right now. And in the case of the Higgs boson, for example, so that theory was proposed over 30 to 40 years ago. There's a subatomic particle that's capable of providing mass. But, because of the limitations of technology at the time, there was no way to pretty much test it or to break down the particles, the atom small enough to discover it. And, they had to wait 20 odd years or so for the Hadron Collider to be constructed at CERN, right? And only after that were they able to test this particular theory. And therefore, the discovery was finally made. So, like you were saying that science was it's the best estimate or the explanation that we have right now. And right now, we are limited by all these constraints. So science will always keep evolving. It will keep changing. The more methods we develop, the more technology we have, it's always going to change. So
yeah, exactly.
But just to address that question,
sure.
I'm not saying that those lower level explanations are bad, because you often need those steps to get to the next step, of understanding.
Yeah.
There's an example. It's a phrase called "lies to children," which is you have a toddler. And the toddler says, "How do airplanes work?" Well, you could go into the explanation of aerodynamics and pressure and all those things, or you could say, well, they have wings and birds have wings, and so things with wings fly. And for a 2-year-old, maybe that's an okay explanation for now, and then later on you can build on that.
I mean, I think actually so we're going to talk about coronavirus now, because it's been in the news all the time. The media absolutely loves this. I mean, look, obviously I'm an infectious disease person, and epidemiology and bioinformatics is kind of my sort of subspecialty within that even. So for me, obviously, this has been very interesting, but the media is really obsessed with this, and it's definitely captured the public imagination, and certainly seems to have been generating a certain amount of fear within the public too. When we look at government responses and the way that people are talking about it online. So I really wanted to give a deep dive into this topic, I think, and hopefully, give some people some of the real science, and also really describe the process, because we talked about the non-peer review podcast. I think that this outbreak had been going on for about 5 days when we first talked about it. We happened to record really early on, and I made some predictions then based on information we had, and all of those predictions were completely off because as soon as we got more information, all of those predictions just weren't accurate anymore. And I think we're going to see here like the importance of information and how actually information has evolved over the last 10, 20 years. Because SARS is obviously going to come up as well. And that was a little different. So I guess in case you don't know, and I assume most people here will know, so coronavirus is an outbreak that started in China. There's a province called Wuhan, which is where the outbreak first started. We know this because there was just a big cluster of cases there. And though there is an outside chance that maybe someone else from outside the region came in and infected everyone there, but really that's the epicenter. We can talk about what an R0 is a bit later, but essentially viruses need a certain density of people to spread. And something like the Wuhan is a quite densely populated area because of the industry they have there, and these like marketplaces where people gather to buy food. So it was a really good little epicenter for the outbreak. And so far, we're up to quite a few cases. So I'm actually looking directly at the World Health Organization situation report. And I actually really recommend that if you're ever worried about something like an infectious disease, we had Ebola a while back, now we have coronavirus, there was MERS. You really should look to the World Health Organization situation reports. They're really easy to get, they're completely public, and essentially there's a day-by-day analysis of what's happening. So right now there are 43,103 confirmed cases, and 2,560 of those are new from yesterday. So that's since the last report, which was yesterday. 2,484 of those newer cases are in China, of which 42,700 are in China altogether. So obviously we can see here that the majority of this is happening in China, and China being a very populated country is unfortunately sort of one of the best places for a virus like this to spread around. Somewhere like India would be another example, or like Tokyo, places that just have a lot of people in a small area, especially for these viruses that transmit through respiration, which of course is one of the best ways for a virus to spread because we all have to breathe and we can't all avoid being near each other, unfortunately. So far there's 1,017 deaths, which is obviously by itself that sounds like a really scary number, but when you think about that as a percentage of 42,000, it's really not too bad. That's less than 1% if my math is correct. Math people, correct me if I'm wrong.
Yeah, it's less than 1%.
Yeah, which look, that's still a scary number, don't get me wrong, but it's definitely not as bad as it could be. We look at things like Ebola that has a death rate, I should say case fatality rate, and I'll explain that word as well because that is important. It has a case fatality rate of somewhere between 40 and 60%, which is pretty bad. If you've got a 4 in 10 to 6 in 10 chance of dying from a disease, that is scary. And one of the things to bear in mind as well, what we're seeing is that most of the deaths and indeed most of the severe disease is in elderly men, essentially. So men over the age of 60 are, for the most part, the people who are unfortunately succumbing to this virus or having more severe illnesses. And this is because, obviously, elderly people tend to have immune systems that aren't as strong as they used to be, and they're at a big chance of having what we call comorbidities, which are other diseases. Diabetes is a big one. Things like colon cancer,
COPD, like asthma, respiratory conditions,
exactly.
Which is actually a pretty big contributor in this case.
Yeah. So these are all things that obviously make you less likely to be able to fight this virus. So I think what I would say at the start, so we don't just bury the lead, is that most people don't have too much to fear from this virus right now. And I think there's this idea in the media that they're always sort of propagating this idea that coronavirus has an RNA genome and it mutates really quickly. And it's true that viruses that have the single-stranded version of DNA, RNA, they do mutate a little quicker. And the reason for that is that they don't have the enzymes to check the quality of the DNA or the RNA in this case. Obviously, viruses need to replicate in order to transmit. And in order to do that, they have to replicate their DNA and all the proteins. DNA viruses tend to have an enzyme that checks for errors as the DNA is made, whereas RNA viruses don't have this, and so errors can creep in. But I think one of the important things to know about viruses is that, one, viruses don't have a brain. They don't have wants. They don't sort of want to make you sick. They don't want anything. And what will tend to happen is natural selection will favor things that make the virus better to spread, not to kill you. And in fact, the two of those are actually normally opposed, it turns out, because viruses that kill, especially kill quickly, they burn out very quickly as well. Essentially what happens is either the population becomes immune, in which case the virus can't spread, or the population kills everybody. So when we talk about viruses from like an epidemiology point of view, which is like the study of virus spread and who it spreads to and where, there's kind of a few stages of a virus. There's an outbreak stage, which is kind of what we're seeing now. Mm-hmm. We have epidemic, which is when essentially it becomes a large problem in one particular area. Then we have a pandemic, and that's when the virus spreads essentially throughout the entire world, or at least to a lot of it. Yeah. And after that, there's two ways a virus can go. And there's a couple of reasons for the first. The first thing that can happen is extinction. Mm-hmm. And there's two reasons this can happen. The first one is that people become immune, essentially. That anyone that would be susceptible has had the virus and survived, or they had the virus and died. And therefore, the virus can't continue to replicate. This has happened through history a lot. The other reason, of course, it can go extinct is that everyone died. And now there's no more host. Which is obviously what we're really hoping is not going to happen here. Mm-hmm. The human race hopefully has some survivability left in it yet. Mm. The other option, though, that can happen is something that's called endemic. And that's when the virus is always there, but isn't necessarily a huge problem, but isn't necessarily not a problem either. So things that were endemic, we have like the flu. The flu is endemic to humans. It just exists everywhere. We can't really get rid of it. But it isn't necessarily the worst thing or the best thing. It exists. And there are a lot of things in humans that are endemic. HIV kind of went through this process of becoming an epidemic, a pandemic, and then an endemic. Currently HIV exists within the human population and we're working to eradicate it, but for now it is endemic. It's just always there. So it remains to be seen really what will happen in this virus. Right now we're at the epidemic stage where there's a lot of cases in one particular area and it's still transmitting. So we're still seeing more cases. And the important thing about viruses is to know what's called this R0 number. And the R0 number is essentially it's like a graph of how a virus will spread. So let's say a virus has an R0 of 5. That means that for every one infected patient, 5 new patients can be expected to become infected. That's assuming a susceptible population. So in this case, that's what we have because no one's had this coronavirus before. No one has the immunity to fight it off just yet. And so essentially everyone is susceptible. So the R0 number will be at its max capacity. The way vaccines work is actually reducing that susceptibility number. So the measles, for instance, that has an R0 of about 16 to 18. Mm-hmm. So that's why measles was such a massive issue in the population, is that for every one person who got infected, 16 to 18 more people could get infected by one patient. Now, luckily, measles is usually fairly mild, and there's like a 1 in 1,000 to sort of 1 in 100,000 death rate for measles. And of course, now we have the vaccine. What happened is that R0 dropped below 1. Mm-hmm. And when a virus drops below an R0 of 1, of course, that means it can't propagate at all because now it has less than one person and there's no transmission chain. So for now, the R0 of this virus is still up in the air. Early estimates put it at like 5, which is pretty scary because that's a lot of spread. But that would also be based on data where there are little clusters. So these are where people like healthcare workers, people like family members, people who would be in extremely close proximity were getting the virus. Now that this has started to progress, I think we're seeing the R0 drop to around between 1.5 and say 2.5 to 3, which is obviously still a fair number of people. That's why we're seeing so many new cases, but it could be a lot worse. The flu, for instance, has a similar R0. And I think the flu is an interesting virus to talk about here because the thing is with the flu is that it has a very similar course of disease to this coronavirus. The symptoms are very similar. You have the fever, cough, the joint aches, and of course it's spread by respiratory routes. And I think one of the interesting things with the flu is that the flu has infected an order of magnitude of like 1,000 more people this year and killed 1,000 more people so far. You know, like the flu kills a lot of people each year and it spreads. But because it's something we already know about, the media doesn't really talk about it as much. But the interesting thing is with the flu is we already have a vaccine. Everyone could get vaccinated. Yeah. And while the vaccine for the flu isn't the best, you know, we all know that the flu vaccine can literally range from anywhere between 80% to like 20% effectiveness, depending on what happens to the flu that year. We do have a vaccine that people can get, but no one's freaking out and running out to get a flu vaccine, when really they have as much chance of catching the flu right now than they do this coronavirus, even in China. I mean, remember, right now in China, it is flu season. It's the winter, which is probably the reason this virus is able to spread. And it could actually be that once we see the temperatures increase and it starts getting to summer, that this virus is less able to survive outside of the body. So one of the reasons that during the winter that you're more likely to get sick isn't because, you know, there's that myth that being cold or wet makes you sick. And that isn't true. But what does happen is during the winter, because the temperatures outside are lower, the virus can survive for longer. And so more viruses in the air, essentially. So, yeah, there's some sort of interesting things. So you flagged this before the show. They've named the virus now.
Yes, so the WHO has just recently given a name for the novel coronavirus that's been affecting us, and they're calling it COVID-19. That's spelled COVID-19. Yeah. And a lot of people will be wondering why the WHO, you know, is coming up with names for the viruses. And there's actually a pretty big social implication here to why we named the viruses. And the main thing is because of stigmatization that's happened. So a lot of people have been sort of associating the virus with China because, as you mentioned, that was where ground zero was, or where the first case was reported. So what happened was people started associating them in a very negative way, saying that, oh, it's the China virus, it's the Wuhan virus. And while it sounds like, you know, it's in another country, but what's been happening as an extension of that is that people of Asian descent, essentially, have been sort of stigmatized as well. Yeah. So we've had people sort of like actively avoiding contact with Asian people, regardless of where they are. You know, that's even been happening here in Australia. And situations like this have actually happened before. So we talked about the MERS virus, and it stands for the Middle Eastern Respiratory Syndrome virus. Right? So when that was a thing, people were sort of actively avoiding Middle Eastern people, you know? And while, of course, you know, there's a lot of climate of fear when a pandemic breaks out, but then, you know, some level of sensibility probably wouldn't hurt people as well, right? Because if not, it just leads to panic eventually. So, and one of these panic has manifested here, like there's this news story on ABC today where a student of Malaysian origin, you know, she's of Chinese descent, but of Malaysian origin, she flew back. She was a student in Perth, and she flew back to Malaysia for a couple of weeks, you know, to celebrate Chinese New Year, I presume. And when she came back, you know, she came back to her rental property where it was just locked off, and there was this huge sign outside. It said something like, "Oh, because you failed to comply to keep in touch with me while you were back home in your country regarding the coronavirus infection, therefore we decided to lock you out of your house." And they actually changed the locks and prevented her from actually coming back in. And now this is a person who's never actually been to Wuhan. She's of Malaysian origin. She's been there for a couple of days where the infection isn't actually at any red level or red alert as far as I'm concerned. And yet the stigmatization has happened, and it's affecting people all around the world. So, the first step of naming it, even though it seems like a very small and trivial matter, is actually pretty important when it comes to the removal of stigmatization. Yeah.
Yeah, it is a big problem. I think that there's evidence that in the States, and bear in mind that the United States has had very few cases so far, things like even just like Chinese restaurants are having trouble because people are avoiding Chinese restaurants to eat.
There you go.
And while it's understandable that there's this kind of fear, I think that the problem is that the media as well has really pushed this out of proportion as well. So it's really hard. I'm just looking at the interactive map now. So far as I know, I actually don't think there have been any cases in Malaysia anyway.
No, if you go further down south, there would have been a few. Yep. So you can see they're closer to Singapore.
Okay, so there's 18.
Yeah. So that is in Eastern Malaysia, and then there is a fair number of cases in Singapore who might have been handling the situation pretty well, actually.
Yeah.
But,
yeah. So yeah, there is a lot of. Definitely people in the comments also talking about the racism as well. And someone who was in China 3 months ago and isn't sick. And I think if you were in China 3 months ago, the chances are that you wouldn't have been exposed as well. But yeah, they're talking about a friend who's there and basically how people can't go outside. And this is kind of an interesting facet of this, because there's been a lot of different reactions in terms of quarantining. Look, obviously, the problem is as well in China that this all happened during Chinese New Year, which is a great time of celebration in the region. People typically come together, and the government's had to essentially ban people getting together in like stadiums and places like that. And unfortunately, that's probably a sensible precaution because of how many cases there are in China specifically right now, especially in those regions like Wuhan. There's definitely some cases around Beijing.
Guangzhou, I'd imagine, being one of the city centers.
So that there are some maps that actually have a province by province breakdown, and of course Wuhan still has the most. And we've definitely been getting unofficial communication by locals talking about how the healthcare situation there is not very good. So because you can imagine the amount of resources this is taking is massive. On top of what is flu season as well, they'd be having a lot of having to isolate patients, but also people with severe disease. They take a lot of care. It takes a massive amount of people resources, physical resources, and of course money. To deal with this kind of stuff. So
they've been building a new hospital. They've been live streaming building a hospital, and it's been going up very, very fast.
I guess that's one of the upsides of having such a huge population, though, that you can leverage those resources to respond to something like this. Compare this to the Ebola outbreak in Western Africa a few years ago now. They just didn't really have the resources at all to deal with this, and the World Health Organization was actually quite slow to recognize the problem. And so I think we're seeing here a different approach, which is really nice. Everyone has really rallied around this situation, and obviously China's offered as much help as they want, essentially, which is nice. And obviously it's up to them whether they want to accept it or not. And so far, I really have to say that the researchers in Wuhan, specifically the virologists there, have been doing an amazing job. I think within a week of this outbreak being reported, they had published the genome of this virus.
Oh, yeah.
And that's massive. And I'll tell you why. During SARS, we really didn't have the kind of technology to do this. And so it was really slow to try and pick up cases. But what having the genome means is that we can create a lot of diagnostic tests very quickly. People have managed to grow the virus in the lab from patients that have come overseas. I think one group even managed to grow the virus from the genome. So they use something called reverse genetics, which is where you take the genome and you package it up in a virus that you already have, and that virus replicates, and basically just from the genome sequence you can create the virus. And obviously I imagine this kind of might give anxiety to a lot of people, but what I will say is, especially someone who works in this environment, and you do too, the precautions that are in place for this kind of work are a lot. I could tell you the amount of training, paperwork that I've done to do this, and rightly so, because everything we do is about minimizing the risk. And the benefit here is that we can develop tests, test drugs even. People are testing drugs on this virus already that may work in this instance. So I think it's really important to sort of to understand how modern science, even from 10 years ago, and 10 years isn't a massive amount of time, but crazy in science, like there's a lot of new technology that exists now that is making the global effort here really important. And I think one of the things to note as well, that I think a lot of the cases that we're seeing be reported is actually about the way cases are reported in this case, not necessarily how many cases there are. So we talked a little bit about the resources that are available. Because these tests didn't already exist, and because it takes a long time, I mean, we've run PCRs.
Yes, plenty.
It takes a day, essentially, to set this up properly. Yeah. And imagine having thousands of these. Now, obviously, each single one doesn't take a day. You can run 300, 400, 1,000 at once, depending on your equipment. But they have this massive backlog of cases that haven't been tested. And the way epidemiology works is that you can't just say people have coronavirus, you have to confirm it. So a lot of these cases coming in are also cases that are just a backlog of testing as well. And I think that's something to bear in mind. So I do want to talk a little bit about the idea that this could mutate, though, into some like horrible pandemic humanity killer. And interestingly, so my honours project was on dengue virus. And what I was looking at during my honours project was how a virus evolves during the course of an infection. So looking at the DNA sequence and seeing how much it changed over the course of an infection. And it turns out that the sequence of a virus changes quite a lot during the course of an infection overall. But what tends to happen is that certain sequences become the dominant population, and they're the ones that obviously replicate better. But what I also found is that certain DNA sequences were completely deleted in a very specific population of this genome, of the virus, in a way that the virus could not possibly replicate. And what was happening is viruses that had all of their machinery still would package this defective virus. And what would happen is this defective virus would get propagated, and it would actually temper the infection. So people who got this defective virus would be less sick. And what happens with dengue virus is it actually turns out that even though dengue virus is transmitted by mosquitoes, people are actually the major determinant in the spread, because mosquitoes have a very short range. It's about 100 meters. And so rather than thinking of mosquitoes as the vector for dengue, what we should actually be thinking about is humans as the vector for dengue carrying to mosquitoes. Because when you track the spread, that's actually how it spreads. You can follow humans to like places where mosquitoes live, and then those mosquitoes infect humans in the area, and then those humans travel. And so there's actually, the reason I bring this up is that in SARS, there's actually a very similar thing where what happened is over the course of the infection happening, a certain gene got deleted, half of the gene, but basically that meant that the protein itself that the gene coded for no longer did anything useful. And this actually, instead of being selected against, this got selected for over the course of infection. And this is a very common pattern in viruses. What we'll see is that as time goes on, this will probably get less pathogenic for a couple of reasons. One, because people will start getting an immune reaction and an immune status. And also viruses just become less pathogenic over time, typically, because it benefits them to have populations that are not dying, essentially. And it's sort of an interesting phenomenon, but this holds true over a lot of different viruses, which I think is worth talking about here. So did we want to talk about the origin? Because that's something that has had a very interesting story so far.
There's been several reports, right? I mean, the most prevalent one I heard, we have heard, is that it's been coming from pangolins.
Yeah.
In fact, another thing that I saw today is that it actually potentially had origins in bats. And if you look at bats as a vector, it's actually happened in a number of like very severe diseases that's happened. So this includes the MERS virus, the Marburg virus, and even Ebola itself. So, and even in the Southeast Asian region, about 2013, we've got a couple of viruses that originated from bats directly, fruit bats, called the Hendra virus, the Nipah virus, and then the Hendra virus, which came indirectly from horses.
Yeah.
So the idea here is that perhaps the virus from the bats has actually spread to the pangolin before it actually spread to humans. Now, it's actually quite interesting to talk about bats here, because bats themselves, being a vector, they have a very interesting immune system. So normally, if you and I get infected by a virus, we would perhaps just generate an immune response, and our immediate reaction would be try to eliminate the virus as fast as possible. But in bats, however, they have a thing where they sort of wall the virus out and form these little pockets to sort of keep the virus in there, where they are actually free to reproduce, but they don't actually affect the bat in any way. Now, the issue is that when this pocket of viruses, so remember that they are actively reproducing and they are actively mutating. So we have this really diverse culture of viruses sitting in these little pockets. So now when the bats either feed, or I believe through their droppings or sheddings.
Yeah, even saliva.
Perhaps, yeah, you're absolutely right. It's actually through the saliva where the infection actually spreads. So perhaps through biting or something like that. And these pockets of viruses go into a host, and then because they've mutated so much and there's such a wide range of them, the probability of having a virus that can successfully survive in the host is a lot higher than one that is just individually mutating at a time. Yeah. So there's this article today that came out on Science Daily that basically talks about viruses that have these bad origins. They are basically more robust, and therefore they are more likely to even jump across hosts as well.
So an interesting thing about bats as well is that they're obviously one of the only mammals that can fly. And unlike birds, who are extremely well evolved to fly, bats are on the cusp of being physically impossible to be able to fly.
Yeah.
And what that means is that it takes them an incredible amount of energy. So a human, the average body temperature of a human is about 37 on average. And in fact, most living organisms operate around 37 degrees. After a few degrees above that, your proteins stop working very well. And actually, one of the reasons we get a fever when we're sick, this is an immune response. And the reason that this happens is that because we're an organism with lots of cells, we can survive being hot for a limited amount of time. But because single-celled organisms don't have an entire network of support around them, they can't survive being too hot for too long. And bats, they routinely, when they fly, reach temperatures above 42 degrees centigrade, I should say, for anyone who isn't using freedom units. That probably means the viruses that are in them, they need to be able to survive 42 degrees, which is obviously, that's fever range. And this probably means that some of the viruses that jump from bats, one of the reasons they can cause severe disease in other mammals is that they actually, we have a comment, use Kelvin like God intended. Yeah, I do like Kelvin. Kelvin's a great scale.
Well, there's no confusion about Kelvin, is there?
No, that's right. Kelvin's great. So I think that these viruses also just kind of can withstand fever a little bit more, which is why we sometimes see these more severe diseases. I mean, it's interesting as well. I think what we will see, and this tends to happen in a lot of bat viruses, is that the bat virus itself didn't come straight to humans, as you said.
Yeah.
So the coronavirus that we have now, we have bat viruses that are 88% similar in terms of their genome sequence, which is actually really high. But the pangolin source is 99%, which is really convincing to me.
Yeah.
On the non-peer review podcast, we talked about the snake hypothesis, and how that was probably not the best research. That's wrong. So the research itself was fine, but the conclusions based on the data were very optimistic on that hypothesis, I think. And essentially what they did is they looked at what they called codon usage.
Okay.
So for people at home, I guess, the way DNA works is it's actually like a multi-step process to go from DNA to proteins to structures. And DNA has 4 letters, as we all know, A, T, G, and C, sometimes U. And each 3-letter combination that exists within that is called a codon. And each codon has a single amino acid. So there are about 20 amino acids that humans have to build proteins out of. And each codon basically has one amino acid. But there are 54 codons, I believe.
I think so.
And what that
sounds
means
about
is
right.
that there are multiple codons for the same amino acid. And different organisms actually prefer different codons. And you can kind of look at the codons and sort of see what
What
species
protein is being formed,
a DNA might be from.
Yep.
But in this case, they didn't really look at enough organisms, and it was biologically unlikely that it came from a snake. But the pangolin makes a lot of sense because even though pangolins are, or pangolin, I'm not sure,
Pangolin. Yeah.
they're endangered species, and it is illegal to hunt and kill them in China, but they are basically, it's a bit like caviar, I guess, like very
It's a
rich
delicacy,
people eat
yeah.
it as a delicacy.
Shark's fin would be another one. Yeah.
And there's an underground market for pangolin. And also, it actually is used, the pangolin, it's actually weird because pangolins are a scaled mammal.
Yeah.
And the scales are used in traditional Chinese medicine as well, which means that these things are around in markets. And I suspect what we'll find out is that a lot of viruses, someone comments in the comment too, they use scales too.
Sure.
What we're finding, I think, a lot with these viruses is that in animals, a lot of animals, their respiratory system, while being mammals still, is actually quite different to ours. And the size of the droplet is really important. Too heavy, and we don't breathe them in. Too light, and it doesn't carry the virus. But I think probably what we'll see is happening here is that the way this virus got into people is that it was actually during the butchering process. Because you can imagine swinging a cleaver at an animal creates all sorts of blood droplets in the air. And even like the lungs and stuff, just fluid goes everywhere. And people would have breathed in the fluid and then gotten the virus. And then after enough introductions, the virus would have basically mutated to be able to infect humans. And in fact, what we already know is that this virus actually needs a receptor called ACE2. So I'll see if I can bring up an image for us because it's kind of interesting how the virus propagates. And basically what happens is the virus needs to sort of grab onto the host cell first before it can replicate. And if we don't have the right type of protein, that just won't happen at all. And so it's kind of one of those things where it's sort of, and I'll see if I can bring this up for us, sort of getting used to live. So I'm going to shrink us here.
There you go.
And here we go. So this is the, yeah, smooth.
Yeah.
So we have this this picture here. So this picture, and we've lost Ben, but we'll get Ben back in a bit. So you can see here essentially and I'll describe it for the people at home who have audio only. These little bits on the edge of here are the edge of human cells, and those little sort of Y-shaped structures are ACE2, and the virus has a protein called the glycoprotein on the outside. That's what gives coronavirus its name. It's all these little proteins on the outside create what's called a halo, and corona means halo in some
Latin,
prestige language.
I'd imagine.
Latin, Greek,
it has
something
to be
like that.
Yeah.
And in order to get into the cell, what happens is that the virus attaches to this protein, and then the cell actually literally takes the virus inside, and the virus can then from there spill its contents into the cell, and the cell's own machinery is what's used to replicate the virus. And we have this protein. So I believe SARS also used ACE2. And MERS used a different protein, which I won't go into the specifics because it's really not important unless you're someone like me. But it used a slightly different one, so that's the reason. So that's why it's able to infect us, because essentially, like, I guess our receptor is similar enough to the one the virus was already infecting for us to be able to see that. So. I guess the last thing I want to talk about then, before we talk too long on coronavirus, is the quarantining that's happening and how...
Hang on, before you get into that, should we make a conscious effort to not call it the coronavirus? If they're gonna be calling it by its new name,
Yeah.
should we?
Well, the thing is, it is a coronavirus, and because it's the one that right now is the one that we care about, I think it's okay to call it the coronavirus. Obviously,
Yeah.
I actually understand your point because that is a bit misleading. There are a couple of coronaviruses that are endemic to humans that cause the common cold.
Sure.
And obviously SARS and MERS were both coronaviruses too. So whichever company here, why was the science white paper pulled that showed 4 inserts from HIV-1? Now, I actually haven't seen that paper. So I'm gonna look this up while I guess so you two are aware of the cruise ship quarantine and the Christmas Island quarantine.
Right.
So do you guys want to just sort of explain that situation while I look up this paper? Because I'm quite curious about this. And we can talk about whether that's a good idea or not.
Well, I actually had a hot take about the naming convention for this virus.
Okay.
So they've named it the COVID-19, which stands for Coronavirus Disease 2019.
Yeah.
Which I think is unimaginative.
It is, yeah, in a way.
And so it also doesn't really, it only vaguely describes what the disease is. So for example, SARS is severe acute respiratory syndrome, which is, as an acronym, it works. But historically, a lot of viruses have been named after patient zero, where they've come from, or the symptoms they cause.
Sure.
So you've got like things like the Black Death, the bubonic plague. You mentioned the Hendra virus, which is from the Hendra area in Brisbane, actually.
Oh, really? Okay.
Yeah, the suburb of Hendra. So the idea of people calling it the Wuhan virus isn't actually that far off historic precedent. For example, the Spanish flu, which is a misnomer because it wasn't originally from Spain, it was from the US, and then it came to Spain through the war, and Spain were just the first people to actually admit that there was, because there was World War One. They didn't want the enemy side to know that they were having a disease. The enemy, Germany, was also suffering the same disease, so it wasn't as though they didn't know about it. So the COVID-19 disease, I don't like because it's also the coronavirus disease 2019 disease.
Yeah, it's redundant, is what you're saying. Yeah,
it's like calling it an ATM machine.
Yeah, I mean,
automatic teller machine.
It's not so much different than calling it either just a coronavirus or 2019 coronavirus. But I guess what they're trying to highlight is the phrase COVID-19. So that sort of just changes it a little. It's just branding, I guess, more than really like a complete name overhaul. Yeah.
Trust me, as somebody who works in PR, I understand branding.
Yeah.
I'm just gonna say that COVID-19 is not good branding.
Yeah.
Yeah, I think the problem is that these names aren't for the public. These names are for doctors. And I should say as well, actually, that COVID-19 is actually the name of the disease that is caused by the coronavirus,
not the virus.
And itself. The name of the virus itself has been
nCoV, right?
So that the International Virus Taxonomy Group, they've named it SARS-CoV-2, so it's SARS the sequel, I guess.
SARS the sequel.
So I'm looking at this paper because this is the first I've heard of the HIV sequences. And so firstly, the paper itself was published on a place called bioRxiv. And something that's important to note when it comes to bioRxiv is that literally anyone can publish research on this, which isn't a bad thing. So for a long time, archive existed. And this is where people would print physics research before peer review. And this was normally to get feedback from other physicists to say, hey, is the math correct? What do you think of the theory? Could we change things? And basically biologists were like, well, that's a good idea. We should do that. And so bioRxiv is a completely open access journal. And
it's like a preprint.
Yeah, so before it's peer reviewed. And so this research was published by a group out of India, it looks like. And the paper itself has been retracted by the authors. So the authors uploaded the paper, and it looks like after comments from people in the scientific community, they have decided that they're going to withdraw the paper and work on it a bit more. So the likely thing for me is that there are probably some mistakes in the analysis, because the DNA is actually freely available. Literally anyone could actually do this. It won't be great television, so I won't, but I could literally run what's called a blast search right now and look for similar genes, and sort of see if there are similarities. The language as well was a little odd. Uncanny similarity of unique inserts into the 2019 nCoV spike. So obviously this was published before the new name, but it's I think probably what happened is that the analysis was possibly a little flawed, and what's happened is that the authors have decided of their own volition to withdraw the paper for now, and they've decided to have a closer look at the data, I guess, which is how science should work. So. Yeah, it's interesting because I hadn't even seen this. One of the interesting things...
As somebody who deals with journalists fairly regularly, I will tell you that no reputable journalist will publish something from arXiv or bioRxiv or chemchive. They all wait until it's being actually published in a peer-reviewed journal.
Yeah. But I mean, I think I actually would like to see the republishing of this. I'd like to see what they did. I think because the paper itself is withdrawn,
but you could actually read the paper.
Not available.
No, it's completely gone.
Because it's withdrawn, you can't read the paper itself. No, I lied. No, it's withdrawn. Yeah. So I would read it if I could, but unfortunately I can't. So actually this brings us on to an interesting discussion point, because what has happened with bioRxiv is conspiracy theorists have realized it's a thing and started publishing their own sort of fake research on it, and then spreading rumors about coronavirus based on the arXiv papers.
So is there no any sort of filter process for getting your article into bioRxiv?
Oh no, because it's completely open. The whole point of bioRxiv is that it's peer reviewed by the community.
Okay.
Which I think is fine, honestly. It's unfortunate that people are using it that way. I think what that means is that as communicators, journalists, and the public, I think we need to do a good job of explaining to people what bioRxiv is and what it's used for and what it shouldn't be used for, which is, I hope, what we've kind of briefly gone into just now. So, yes, I think... so that's interesting, actually. So thanks for bringing that to our attention. I actually would be interested to see if they republish with new data and new analysis, because, I mean, if it does turn out to be true, that would be obviously very interesting. I find it unlikely that it could be true, because HIV is a retrovirus.
Yeah,
and coronavirus doesn't do that. It doesn't have a retro... the RNA is directly made into proteins. If I actually bring up the thing here, you can actually see what happens there. The RNA is directly made into proteins, which is made into new virus. And so, I think it's unlikely that HIV... And obviously, I'm guessing the inference or the sort of the conspiracy here is that COVID has been, or SARS-CoV-2 has been engineered somehow with HIV proteins.
Yeah.
But I don't think HIV... They talk about the glycoprotein there. And I don't think that it's actually the same... Yeah, the spike protein. So the spike protein of COVID actually binds to a different receptor than what HIV binds to. So this Twitch is making me feel uncomfortable, as I'm having to admit biologists may actually be smart. Said the true physicist. Well, yeah, hopefully. Oh, question. The other week on the news, they were saying that coronavirus could have originated with snakes. I recently heard secondhand it was withdrawn. Is there a good place where I can get news about papers being withdrawn? So actually there are a few. the best website is called Retraction Watch, but they tend to focus on papers that are withdrawn for sort of dodgy reasons.
Right.
QUT featured in that once, actually.
Oh, okay.
Best not to discuss that with colleagues, but that did
not
happen.
yet. No.
To be fair, almost every university has had a Retraction Watch moment.
Yes.
Because scientists are human, and sometimes scientists either make mistakes or do dodgy things, unfortunately. So I believe there's actually a really nice website that's keeping track of coronavirus. There's like a little microsite that is looking at all of this stuff, and they probably would talk about the retraction. Unfortunately, it's really hard.
It wasn't a retraction though. It was just under new information. It kind of, you said it yourself, Ross. The research was good. Just the conclusion was probably optimistic or stretching too far.
Yeah.
Speculative, perhaps.
So, unfortunately, when research comes out, it's widely reported. When research is withdrawn, it's barely ever talked about. So, one of the interesting things to talk about is how there was a case where a Chinese businesswoman went over to Germany and a bunch of people in Germany got infected. Mm-hmm. And that was used as evidence that the virus could be spread asymptomatically because the Chinese businesswoman didn't have symptoms at the time. And that was published in the New England Journal of Medicine, probably the best journal in the world for medicine. Now, it turns out that they actually didn't get to talk to her directly. This was based on secondhand reports, and that they have since actually tracked down this woman and talked to her, and she was sick. But we haven't heard much about that in the news. All we heard about is that, oh my God, it's spreading asymptomatically. So, unfortunately, I actually don't have a great answer for you, because there isn't a great way of tracking things that have been withdrawn, really, except for looking at places like Nature, Science Mag, which is the science magazine. There's also a website called CIDRAP, and that is an excellent website. And they have a webpage dedicated to talking about the latest situation when it comes to coronavirus research. And actually, because we're on Twitch, which is amazing, I can actually just post this link in the chat, and everyone can have a look. And it looks a bit empty right now for some reason, but there should be. Let's have a look at novel coronaviruses. Maybe this is a better one. But essentially, here we go. So CIDRAP is great because they look at all of this, and they're likely to talk about withdrawals as well as new research. And so this is the Center for Infectious Disease Research and Policy from the University of Minnesota, of all places. So I guess that's coronavirus. We've talked about it for a while, but hopefully people are informed, because it might affect us. That's the thing. Like in Australia, we're lucky that a lot of us here are younger and probably won't. If we got infected, we'd have a flu-like episode and we'd be okay. But we really don't want this to become endemic, because it's really another virus that we have to then get a vaccine for, develop good drugs for, etc. And I think that what we should be focusing on is obviously trying to give China all the resources we can. I mean, I think it's obvious that China are struggling, and I think that's obvious because of how many cases they're having. Any country would be struggling. If we had the number of cases that China is having, our entire economy would have collapsed.
Yeah.
Absolutely. Australia only has 24 million people. If we had 42,000 cases in less than a month of a virus like this, with like 7,000 severe cases, there is not a hospital in Brisbane that could deal with that. Not a single one.
No.
This is a big deal, but I also don't want to give people the impression that there's some apocalypse about to happen, or that we should stop going to Chinese restaurants to eat food.
Yeah, absolutely.
I go to UQ a lot, and they have a lot of Chinese international students. And UQ, there's a couple of universities in Australia that actually delayed the start of the semester. And I actually think that's not a bad idea, but not necessarily because I'm afraid the virus would spread, although that is obviously an upside. But a lot of people are still trapped in China, and I think delaying the semester actually helps these international students get back to Australia in time. So you don't need to avoid Chinese people. and if you live in a Western country, chances are that Chinese person you think you're avoiding isn't even from China anyway.
Yeah.
So
And if they are, they probably haven't been to China recently either.
Exactly. Like there's a lot of travel from Wuhan, but the vast majority of Chinese people, or I'm going to say it, Southeast Asian people, because white people obviously aren't exactly the best at recognizing people from other places in the world.
Well.
They're probably not even from China. So I think that you should keep going to your local Chinese restaurant. If you have Chinese friends, obviously don't avoid them. That's really mean. We'll see. I have a feeling we'll be talking about this, hopefully in a little less detail, but in 2 weeks' time. We shall see how the Christmas Island endeavour has worked out in that time. I mean, we should very briefly mention the cruise ship in Japan. Because I believe at the moment 78 people are now infected with coronavirus on that ship. And I don't think that was a great idea, personally. I don't think quarantining people on the ship was necessarily sensible. So it's hard because quarantining is known to work. It's a really good intervention in situations like this. But I think putting people up, putting people in a very small space, not unlike an incubator, say, or a petri dish.
So your argument is more on the fact that they got quarantined on the ship rather than anywhere else in the universe, basically.
Look, I don't know. Obviously, it would cost money to put these people in like hotel rooms and separate them from each other. But a lot of the staff are becoming sick now because they're the only ones who can go room to room delivering food and stuff. I think time will tell whether this was a good idea. I think in theory the best solution is home quarantine. But the problem with that, of course, is that not everyone will voluntarily stay at home. But that is the ideal thing for people to stay separated.
It would need some
at home.
level of enforcement, I guess. Like in China, they are enforcing, home quarantine in a lot of cases, and they have implied curfews and things like that. But they also, I do believe that they have people that are enforcing it, either the police or the military, will probably be supporting in that role.
And China's kind of uniquely suited to be able to do that, given that they do have a large, police force and sort of their state resources are skewed towards enforcing something like this already.
And the other thing is that they've had so many of these outbreaks in the last 10, 15 years that they already have the systems in place. Like the moment this COVID-19 came out into the news, their response was instantaneous. The hospitals were already responding as fast as they can. Everybody went into home quarantine. And the people themselves, they were very compliant when it comes to this because they understand the implications as well. So they've been actually doing a pretty good job, if you can think of it. It could have been a lot worse.
Yeah.
Yeah.
All right. Well, I think that will conclude coronavirus talk. We went for a long time on that one topic. But I think having this like one large discussion now will be useful for people who do want to learn a little bit more. And hopefully I don't just want to... I hope that we've kind of taught people a little bit about epidemiology and outbreak science in general, and that hopefully... I think the aim is that we can give people information that you might actually be able to use in the next one. Like there could be another... I mean, I say there could be another Ebola outbreak. There is an Ebola outbreak right now in the Republic of Congo. So these things happen, and these ideas, they can be used for any situation, like the flu, for instance, like... All of the things that I would recommend to protect yourself from coronavirus, and I know I keep trying to wrap up, but actually this is something useful. If you want to protect yourself, I don't think wearing a mask is especially helpful. I'll say that outright. Masks are really good at stopping sick people transmitting virus to healthy people. They're a lot less useful at healthy people avoiding getting sick. There's a few reasons for that. Firstly, the masks themselves, they aren't necessarily designed to filter out the fine droplets where the virus particles are spread. Whereas masks that, when you expel the air, the droplets start off a lot heavier and they get sort of captured. That's why surgeons wear the masks, because the masks are really nice at doing that. Obviously, I think the main thing that you can do, the other reason actually I should say that masks don't work often is that you still end up touching your face.
Yeah.
Like you touch your eyes, your mouth, your nose. These are all what we call mucous membranes, and they're like Yeah, they're all direct conduits to your body, essentially. That's how a lot of viruses spread. If you want to stop yourself from getting sick, wash your hands and wash them frequently.
Number one rule.
Every time you see a sink, wash your hands.
Yep.
We're lucky we do that because of our job.
Oh, absolutely.
I wash my hands at least 10 times a day.
Yeah.
And more, even when we're at
Probably
home.
more,
You know. But we have this drummed into us, and this is what I'm talking about when it comes to training and stuff. We're trained to do this stuff. But wash your hands often. You know, hygiene is really the most important thing. If you need to cough, ideally you would cough into fabric. But actually the dab, weirdly, is one of the best things. If you don't have something to cough into, you can literally cough into the small of your elbow, and that actually contains a lot of the contains a lot of it. So please do those things. Wash your hands often. Try not to touch your face. That's just good life advice in general, actually. Try and contain coughs into your hands, your elbow, or even, it sounds gross, but just into your shirt. It doesn't look great.
And now you're infected.
Yeah, it doesn't look great, but it is good. All right, so I don't think we're going to have time to get to a lot of the stories that we had planned outside of coronavirus.
Yeah,
but we brought Ben all the way over the internet to join us today, and Ben had a cool story to talk about. So why don't you tell us about your story, Ben?
Thank you very much. So I am going to present this story to you in much the same way that I present the stories on the non-peer-reviewed podcast, by which I mean I know what I'm going to say, and neither of you know what I'm going to say at all.
Okay.
So we're going to get some live reactions from you both as I explain this cool bit of science story. So I have this here, and we're going to start off. Rare European cave salamander stayed in the same spot for 7 years.
My only question is, was it meditating? Was it sort of a wise person in its community?
So many species of salamander live surprisingly long lives. But among these enduring amphibians, there is one outlier called the ohm, or Proteus anguinus. It has been well documented that these small white cave-dwelling salamanders can live well into their hundreds, but scientists have now gained new insight into the creature's glacial pace of life. In a study which makes sloths look recklessly hyperactive, divers documenting the movements of ohms in the Herzegovinian caves in the southern region of Bosnia found that over a decade, individuals tended to move less than 10 meters. However, one extraordinarily inert individual was found not to have bothered moving once in over 7 years.
Wow,
To be fair,
That's amazing.
To be fair to these slimy, creepy crawlies, as they're known to the locals, they're not highly gregarious. They have no predators. They are highly resilient to starvation, able to go without food for several years. They're blind, and they live in complete darkness underground and underwater. So they have apparently they only really move in order to mate, which they do on average once every 12 and a half years. And
And
yeah, in the caves in which they dwell, food is typically scarce. But when they are able to, they feed on small crustaceans such as small shrimp, snails, and occasionally insects. So very, very sedentary lifestyle.
And how long do these salamanders live for? Just out of curiosity.
Hundreds of years.
Hundreds of years, yeah. Okay. And they in a span of 100 years, one salamander would move less than 10 meters for the entire lifespan, basically.
Yeah, so
something like that, if my maths is right. Yeah.
On average, they move about
a meter.
1 meter a year, or 10 meters in a decade. So maybe there's like 9 years of not doing a lot, and then suddenly, that 12 and a half year mating cycle comes up and they go, time to do this.
Time to move. Okay.
So when
2
you
meters
say
that way.
when you say move 10 meters, do you mean like in terms of territory, or do you mean literally if you counted their entire like
Movement,
footprint, like if you calculated their entire movement, it would only be 10 meters?
So
Or
what the divers did was, in much the same way that parking inspectors come along and chalk your car, your tires, to see if you've moved, they marked where the salamanders were, went away, came back, weeks or months later, marked it again, and just saw how far they had moved.
Oh, we're getting a physicist type question
no.
now. 10 meters vector or scalar?
It would be vector, I would say. 10 meters in a direction.
Yeah.
So, I mean, being the animals that move
Actually,
such a short
scalar.
distance.
Scalar.
Oh, there you go. Yeah, sorry. I what I wanted to ask was, so these species of salamander, are they just found in a specific region, or are they quite widespread? Because they're moving only 10 meters.
No, so the Olm are not particularly widespread as a species. They are cave-dwelling salamanders, and like many cave-dwelling species, they are only kind of found in small pockets.
Mm-hmm. And just within the southern region of Bosnia, and it hasn't been discovered, to your knowledge at least, anywhere else, or?
Not from the reading I did for this article.
No.
Yeah.
Yeah.
Man, it's insane, isn't it? Because like, when you think of living creatures, you think about movement a lot. I mean, we're a fairly energetic species. We're surrounded by things like insects and things that fly around, but there's a large amount of life that really does just kind of find a spot and stay there for a long time.
Well, like here in Australia, we have koalas, for example. But they were told to sleep about 20 hours a day. And then for 2 hours, they get up, feed, and then go back to sleep. Or something like that.
Well, if you move outside of Animalia to the other kingdoms of Tree of Life, fungi and just plants don't really move a lot.
No. Yeah. But then I do believe they pretty much evolved systems that allow them to survive in place. Like, for example, the magic of photosynthesis. And a salamander, well, at least not to my knowledge, are able to they have the capacity to perform that,
Especially these ones because they're underground.
Yes, exactly. There you go. So
they would have just very, very slow metabolisms.
Yeah.
There is a somewhat dystopian future for humans in which VR becomes so good that we could turn into those salamanders, right?
Oh, you're talking about the ending to the movie WALL-E?
Yeah, basically.
When they discovered those humans flying around in pods and basically drinking liquid pizza or something like that. So we're definitely heading in that direction, I'm pretty sure. But
yeah, man, I just
so there we go. That's a sample of some of the stories we cover in the non-peer-reviewed podcast. Yeah, it's a lot of fun because the discussion tends to be like quite natural, obviously, like this, because we sort of like get back and forth.
We get bombarded with science, essentially. We get confronted with science, and then we have some pretty good discussions after that. So it's a lot of fun. Now I think you had a story, Tina, as well about some new ways of diagnosing, which is quite interesting.
Yes. So I guess this sort of relates back to the whole coronavirus thing. But anyway, the story goes is that some researchers over at the University of Cincinnati, they've developed actually a portable laboratory that sort of connects to your phone and then enables you to diagnose diseases that you have on the fly. So how it works is you just take this little plastic chip and you pop it into your mouth. So it just sort of makes contact with your saliva, which then gets pulled up by capillary action into the chip itself. And then you take that chip and you pop it into this little credit card size box, right? And inside the box, they have basically freeze-dried antibodies and luminescent particles, which basically help to visualize either if you have certain types of proteins or even hormones that is elevated in your body. And then it transmits all the data over to some doctors in a practice, and they are able to perform to observe the results and basically conduct a diagnosis on the fly. So this is pretty amazing for a couple of reasons. So one being is that you basically could perform a diagnosis without actually making the effort to go to the clinic, right? So when you talk about situations like the coronavirus outbreak, where everybody's almost confined to houses, if you needed to report that you would have a disease or would you have the infection, this is really a surefire way to do it. It's quick. It doesn't need you to necessitate you to leave the house and potentially become a risk of infection to anyone else. And it just places less burden on the clinics itself, which is fantastic. And of course, the faster we get diagnosis done out, we can more quickly, administer the cure and therefore control the disease and preventing it from going out of control. So this is a pretty fascinating piece of technology when it comes to disease control in general. But another important aspect of this particular device is that it is able to actually diagnose conditions such as depression. So these are mainly hormonal based stuff. And why that's important is that, mental disorders in general, really, because for one thing, people who have mental disorders like this, there is a sort of difficulty for them to sort of seek help and actually leave the house. So it's a question of accessibility, because not everybody is willing to talk about stigmatized conditions such as this. And even this applies to sexually transmitted disease such as HIV and things like that, because you do not feel comfortable speaking about a situation like this, provided you have it. And therefore, this particular device is able to bridge that gap and make treatment for this a lot more accessible. And
So this is kind of like the self-service aisle of medical devices.
Pretty much, it's the cool express checkout for yourself.
I can imagine a future though where you literally you feel a bit sick and you go to the pharmacist and you buy the little cartridge and you plug it into your, these things. Yeah. I actually didn't bring it with me and I will one week, but I have like a sequencing device that's literally smaller than my phone now. Yeah. And then it gets sent off to a GP, and if you need to go see them, you do. But if you're kind of probably fine, you just go on about your day or whatever. I think the idea of like the being good for rural people is really good because because these are freeze dried. What that means is that they'll be incredibly stable, and they would have a long shelf life, you would think. And so what you could do is ship them out to rural places and just have them available. And let's say you lived on a farm like remotely, like
if you're like 20 kilometers away from the nearest hospital, and you'd have to drive if it's anything. So
there are some people in Australia who would have to drive for a day to see a doctor if they were sick. Having something like this would be amazing. You could have a couple of these in your medicine cabinet, and that'd be it.
The hardest part would be the connectivity with whatever device you're using.
Of course.
So, for example, in very, very rural Australia, out in the middle of nowhere, they have these. They're kind of like, they call them like internet towers or phone towers, which are basically like a satellite dish that you stand in front of, and you put your phone in one, like on like a resting place, and in that one location you can get a signal. So even like that would be useful in those kinds of situations.
That's for sure.
But I guess like the medical technology is great, but it's how it integrates with the other systems is the part that I'm a bit iffy about at the moment.
So I guess the particular objective of this one was more rapid diagnosis, and it just eliminates alleviates the burden. But then what you're probably addressing, and it is an important one too, because accessibility to healthcare is a serious problem. We've seen conditions such as asthma, for example. I went to a talk last year where they talked about morbidity of asthma here in Australia. And about 30% of the fatality from asthma, because we know it's not really that fatal a disease. You can manage it pretty well. But the reason why about 30% of people died is because of accessibility. They were too far away from a clinic. They didn't have access to get advice over the phone even, and they just pretty much died because of that. So having like what you're addressing, the accessibility to healthcare is the definite way, direction we should be moving in, for sure. What else? Is there a couple more topics shall we talk about? Or
so I guess we'll end it on a sort of a nice fun one.
Sure.
So a few weeks ago in January, unfortunately, Voyager 2 went offline. And obviously, Voyager 2 is one of these crazy pieces of human engineering that has been somehow operating in space since the 70s, I believe.
Yeah, 1977 when it was
Yeah.
first launched.
And
Huge
Voyager 2 is one of my favourite spacecraft of all time because one of the people who worked on that is kind of one of my heroes, Carl Sagan. So Carl Sagan, of course, probably a lot of people, our age wouldn't really even know who he is, but in the 80s, in the 70s, 80s and 90s, Carl Sagan was a science communicator, and he was a very he was very optimistic about what humanity could achieve. And he was one of the first people to like bring science to the masses. So he was the original host of Cosmos.
Oh yeah,
which of course Neil deGrasse Tyson recently has done a remake of. So he's an amazing guy, and basically. Yeah, so the comments are kind of preempting the story here a bit, but Voyager 2, the reason that I really like it is that one of the things that Carl Sagan did, as Voyager 2 was zooming out towards the edge of the sort of planetary solar system past Neptune, you should know that it takes an extraordinary amount of resources to maneuver these probes at the speed that they go at. And he persuaded the people in control to essentially 180 the probe, to point the probe to look back at the Earth. And he captured the picture, and it's called the pale blue dot. And I'll find it because it's really worth putting up on the screen. It is one of the most humbling pictures of all time. Because what it shows is a picture of the Earth from space. And in that picture, Earth is a single pixel.
Yeah.
And one of the cool things that Carl Sagan would go on to talk about in Cosmos is that literally every person on this planet that ever existed and that ever will exist literally exists within this one pixel in the universe. And Lord Ignator has a cool article about that now. So I'm just going to shift this over just to why we talk about this so that we can show the picture of it. But yeah, so that's the pale blue dot. And again,
maybe could you zoom in a little to make the image a little? No.
Yeah. Ben is again missing, unfortunately.
Yeah.
So I'll bring Ben back later on. But yeah, so this is the picture, and you can see the little dot there. So yeah, that'll go away. So the good news is though is that it's back online. So they basically what they think happened is one of the systems messed up essentially.
Yeah.
Because obviously Voyager only has a certain amount of power, and it kind of, in order to stop itself from overheating or for too many instruments to get broken, if a lot of things happen at once, it actually powers itself down. And the cool thing is that despite the fact that Voyager is now actually outside of the immediate influence of our sun, which is a massive, massive amount, the volume of space, it's not even comprehensible.
Yeah.
You know, like we have the planetary part, and I must admit I'm failing a bit because I don't really know what that bit's called, but like the inner solar system essentially, the bits where the planets are. So Neptune kind of sparks the end of that.
Yeah.
Then you have like the Kuiper belt and the Oort cloud, and that's a massive cloud of objects in space. And then beyond that, you have the heliosphere, which is kind of like the immediate influence of the sun. So like the sun's gravity, the sun's solar wind and stuff. And then once that kind of peters out, then you're in interstellar space. And each part of that system is like orders of magnitude bigger than the rest. Like the amount of space from the sun to Mars is completely nothing compared to the amount of space from like Mars to Neptune. And the amount of space from Neptune to like the Kuiper belt or whatever is again huge. And the difference between the Kuiper belt to the Oort cloud is
exponentially
absolutely
larger.
huge. So you can't even imagine the distance. So it takes 17 hours for the communication to reach Voyager, and then another 17 hours for that communication to come back. And to me, it's just crazy that they managed to reset this relic from the 70s.
Yeah.
I mean, how many computers from the 70s are still operational? You know, like some IBM thing from the 70s. Virtually none of them, right? The ones that ran on little tubes, literally vacuum tubes and tape. Whereas this machine that we built as a species is somehow still there. And it's kind of sad, right? Because you look at stuff that's happening at the moment in world politics, in sort of, especially Anglo countries, we managed to come together as a species to achieve this, and yet we still can't feed people or like guarantee people a roof over their heads. It's just insane to me that we have it. Oort cloud, still not proven, is outside the heliosphere. There you go. This is why I love having an audience, because there's always going to be someone smarter than us on pretty much every topic that we talk about.
Yeah, number one rule of science. There's probably someone smarter than you out there.
Yeah. So thank you for that comment, Torgo. So the Oort cloud is outside. So I'm guessing then, I'll ask Torgo, who's in the audience. I believe that Voyager has left the heliosphere, but I guess technically then hasn't left the Oort cloud yet, because if the Oort cloud is outside the heliosphere, there must be a lot of that left over.
Yeah, he's correct. Well, they are correct.
Yeah.
So as somebody with a physics background and a love of space, so the heliosphere is the protective bubble that our sun generates, which is full of charged particles to protect. It generates it magnetically. This big magnetic field to protect us from interstellar charged particles, and it crossed that barrier a number of years ago. So I want to point out something interestingly. So when Voyager 2 launched, it was generating 470 watts of power.
Yeah.
So think about not so much now, but maybe 10 years ago, our light bulbs were like 60 watts.
My computer, my computer at home requires more than 470 watts.
Yep.
Yeah. So it uses that power to heat itself, so it can actually run up. Because if it runs out of power, it can't actually heat itself, and it would literally freeze. The computer would freeze because the machine would freeze. And what happened with the shutdown was, it was running 2 operations at once, and the safety system realized it was drawing too much power, and it shut down those operations, went into like standby safety mode, because the power is a radioisotope. It's a radioisotope thermoelectric generator. So it uses radioactive decay to generate heat and power. And so it may have started with 470 watts, but it's the half-life is about 87 years, so it's almost half what it launched with. And so eventually, it's just because of radioactive decay, it's going to run out of power. And unfortunately, that's probably going to be within this decade. Probably within 5 years, it'll run out of power.
Yeah. Because of how half-lives work, like the amount of power it has available to it now would also be significantly decreased compared to when it launched, right?
Yeah.
And I believe...
Also, something interestingly, when we send a signal to Voyager, any spacecraft in deep space, we have to actually aim where that spacecraft is going to be.
Yeah.
Because it's a moving target, and we are sending that radio signal to intercept where this target... Like we are... Because radio signals are traveling at the speed of light. At the speed of light, we have to aim where this thing is going to be, and then aim to hit it. And then when it transmits back to us, it's moving and the Earth is moving, so it has to aim to where we're going to be as well.
Yeah.
It's like playing frisbee on moving trains traveling past each other.
Yeah. And the amount of, that signal isn't strong either, right? So there wouldn't be a lot of leeway in terms of like where that signal could actually be picked up from.
Yes.
So we have a comment here. Isn't the Voyager craft in low power mode? I thought most of the instruments were turned off after its primary mission was completed to conserve power. Yeah, I believe I've heard that as well. I think that's true.
Yeah.
So they're only...
It is running in low power mode. It's only running 1 or 2 instruments at a time at the moment.
The magnometer looks like one of those.
Yeah.
And another thing I read today is that they actually have already, because of the power requirements and the decay of the plutonium core, essentially, they actually had to turn off the heating systems for most of the instruments. And they were quite surprised the fact that even with it turned off, the instruments have actually been running for the past 10 years or so, even after the shutdown. So yeah, that's a testament to the build quality from the 1970s, I guess.
Well, part of that as well comes down to heat doesn't radiate away in space particularly easily.
Right. No conductivity from anything. Is that right?
So there isn't atmosphere to draw the heat away, so it has to radiate through it's called
Infrared,
black body,
basically.
infrared. Basically, it just has to generate.
Right.
So yeah, long live Voyager, I guess.
Yeah.
Yeah.
And it is a sad thought because last year we lost the Mars rover, I believe. It went into a shutdown mode. And based on the estimations of the plutonium core of the Voyager 2, it's only a matter of time before we see that. And this is the longest traveling probe, I do believe, compared to the 5 that we have launched into space. Yeah. And only one of 2 to actually reach the what they call solar escape velocity. Is that the right phrase, Ben? You can. Yeah. So yeah, it is a pretty huge deal, and it'll be a pretty sad day when the Voyager actually does shut down. And
Well, eventually they'll be overtaken by the Pluto probe that went by Pluto a couple of years ago, because it's traveling faster than they are. So eventually it'll overtake them.
Because the Pluto probe did a lot of like gravitational gymnastics, didn't it, to reach the speed? And it has a drive on it, right?
Yeah, well, both the Voyagers did as well. They had this fantastic alignment of all the planets, so they could go one to the next, 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 in something they called the Grand Tour. And what happened was when they got to Saturn, one stuck around for a bit longer, and the other one slungshot its way out to Neptune and Uranus. So they're both they're going very, very fast. But as we build new spacecraft, they will eventually be overtaken by the next fastest thing.
Yeah, which is pretty cool. And now we have like ionic drives and things as well, which actually provide a lot of velocity, right?
Yeah, so ion drives are great because you don't have to carry much of a fuel source. They are basically just electrically powered. And they propel themselves by firing electrons.
Yeah, which is really cool. Like, it sounds very futuristic, but this technology has actually existed for quite a while. It's just we're only just now starting to really implement that. So yeah, actually, this is kind of a nice note to finish the story on from Torgo. So it was designed to reach Saturn in 81, and reaching Neptune in 1989 was considered a bonus. So
and it's still going, to who knows how far at this point. Yeah.
Which one melds with space AI and which one gets blasted by Klingons? Well, the Klingons are all the way in the Alpha Quadrant, the Beta Quadrant, right? So that could take them quite a while to get there at the current speed.
If I remember my Star Trek movies, one of them becomes a super intelligent and eventually comes back towards the Earth in a giant spaceship.
Yeah.
That's the AI.
Yeah, they definitely find both of them in Star Trek. In the Star Trek canon, both Voyagers get discovered at one point. I'm pretty sure. So, all right, so folks, that will mean the end, unfortunately, of the Science Club show. Thank you to everyone who tuned in live. It's been great having your comments. Normally, we will run a question section where you can just ask science questions, but we ran quite a bit over time with all of the coronavirus chat. So, but yes, twitter.com/Science Club Show. So if you ever want to send a question ahead of time, obviously we'll research the question and use the section for it. But you can always post your questions live as well. With 3 of us on there, chances are one of us will know the answer, or we can at least sort of fill in the gaps. Thank you very much for joining us. Thank you, Ben, for joining us via the wonders of the internet.
Yes, thank you for having me on the show.
Thank you, Tina, for agreeing to delay dinner till after the show.
Well, it was a great experience, so I'm so glad we did this.
Yeah, and thank you most of all of you folks. So yeah, for everyone who gave us a follow, thank you very much. At the moment, we'll be going every 2 weeks. So I guess the next one will fall on the 26th of February. And it's Ben a Jedi. He looks like a Jedi.
He could be a Jedi. Yeah, he could very well be a Jedi.
The thing is, I am bad at mind tricks.
Yes.
Ben's bad at mind
And
tricks.
also check out the Non-Peer Review podcast. You can find the Non-Peer Review podcast on Twitter, on Facebook. It's a really great show, and I have a lot of fun. That's a podcast. And for anyone who wants to listen to the show again, I'm not sure why you would want to listen twice, but we'll be putting this on YouTube and as a podcast too, so that you can get it as many ways as possible. And yeah, we'll be back in 2 weeks. Thank you very much.
Thanks for having me on the show.